So our research is with Sanfilippo syndrome type B. We are trying to develop a neural stem cell gene therapy to treat the disorder. Due to the complications of, the lack of success with enzyme replacement due to the blood-brain barrier, and even the kind of lack of success with hematopoietic stem cell transplantation, we've thought of trying to introduce our therapy directly to the brain using a neural stem cell system. So the goal would be to take autologous cells, reprogram them to induce pluripotent stem cells, differentiate to neural stem cells, perform our gene therapy, and transplant. So we're kind of doing a proof of concept and modeling that in the mouse system that we have. So we took mouse embryonic fibroblasts, we reprogrammed them to induce pluripotent stem cells, differentiated to neural stem cells, used a lentivirus to upregulate NAGLU in these cells, and then we transplanted them into the brains of mice. Right now we have a long-term study going, but the one that we've processed so far was about 9.5 weeks after transplantation. The engraftment was okay. It wasn't as solid as some of these other published neural stem cell papers, but what was kind of surprising is with the amount that we got, we showed these marked reductions in LAMP1, extreme decrease in GFAP, so these decreases in astrogliosis, microgliosis, and the lysosomal homeostasis. So it's really promising because this is just after like our second or third try doing transplantation. So we're kind of fine-tuning the neural stem cells to get them to engraft a little bit better. And we imagine that after we improve our engraftment by 20 or 30%, we'll have almost a complete reduction of these disease markers. The mouse model has a truncated version of NAGLU within it, causing it to have almost zero NAGLU activity in the assay. We are using lentivirus, which integrates kind of randomly across the genome. So the downside of this study is that we're not using an endogenous promoter. We are at risk for oncogenic effects. But as a proof of concept to show upregulation of NAGLU can not only be secreted and uptaken by deficient cells, but if we can get a certain amount of NAGLU in, we can get the brain pathology to go down. So we are using a human cDNA construct in our lentivirus. So it's a human protein in a mouse. We haven't seen any immunological effects from that, but those are the basic genetics of what we're doing.